वो तो मुझे नहीं पता है हिमानी. Maybe very soon, because physiotherapy students are going to join. So hope that we are able to see each other face to face very soon. So hope for the best. When we say sterilization and disinfection, remember they are very, very basic concepts and very, very important in patient care. And in microbiology curriculum, they are definitely very important. So today I'll be talking something about sterilization, and then next week Dr. Nemika will continue with the same topic. So when we are talking about sterilization, I'm sure you do have some idea about what sterilization talks about. Can I get one or two comments about sterilization? You can write in the chat box, or you can unmute and say, what is sterilization? What do you know about sterilization? No idea? What is sterilization? Any idea? Khyati, can you unmute yourself and tell me what is sterilization? What do you know? Just in case if you don't know, that's okay. But can you just unmute yourself and say? Are you there, Khyati? Akshata? Are you there? Muskan? Login करके गायब? That's not the right thing to do, beta. Okay. So you know that it destroys all microbes, can clean a thing by killing organisms. Okay. So let us see what I have to add to it. But I'll request when you are attending classes, it is good to get involved so that you actually gain something out of the classes. So what we are going to talk about today in terms of sterilization is get a clear concept about some very commonly used terms. You come across terms like sterilization, disinfection, antisepsis, asepsis, decontamination, cleaning. These are different terms which are used in patient care. So you, as a scientific person, you need to differentiate what exactly is the difference or similarity between these terms. And we will also talk about few physical methods of sterilization. Chemical methods, maybe you'll learn in the next class. Okay, so when we talk about this sterilization, please read the difference between sterilization and disinfection carefully. I'll read it for you. Simultaneously, you can also read. So it's a process by which all living microorganisms, including viable spores, are either destroyed or removed from an article, surface, or medium. So this means you are making a surface, article, or medium almost completely free from microbes, right? So the reduction in the number of organisms is at least 10 raised to the power 6 log colony forming units. So when you are trying to say a thing is sterile, that means the thing is free from almost all kind of living microorganisms, including spores. When you say disinfection, it is a process that destroys or removes most, if not all, but it does not really target the bacterial spores. So can you see the difference? The reduction in disinfection is 10 raised to the power 3 log CFU of microbes. So something sterile means almost free from microbes. Something disinfected means you are reducing the load of microorganisms on that particular surface or article or in the medium. You are not ensuring complete absence of organisms. And disinfection process targets vegetative organisms, the vegetative cells, growing stage. When they are in resting phase, that is, spores are not targeted when you're trying to disinfect an article or a surface. So disinfection is trying to reduce the number of microbes so that even if there are some pathogenic organisms present on that particular surface, the number of those pathogenic organisms is not going to be enough to produce disease, right? But sterilization means complete absence of organisms. You don't want even spores there. Keep the difference very clearly in your mind. If there is any confusion, feel free to unmute and ask. Then you use the term asepsis. Asepsis. Asepsis means you are just, yes, Heth, what do you want to ask, beta? What do you want to ask? You have raised your hand, Heth. Nothing? Okay. So when you're saying asepsis, that means you are using something on a live body surface. Asepsis is also a process of disinfection. Please remember. But here you are using some chemical agents, which we term as antiseptics, and you are using them on live body surfaces. So when you mop up a tabletop where you are working in the laboratory, you will call it disinfection. But when you clean the skin, you can call it asepsis also. Though you also use the term as disinfection of skin, that's fine. But remember, all disinfectants are not safe to be used on living tissues or surfaces. So when you are using asepsis, keep in mind that you are trying to inhibit or reduce the number of organisms present on skin. When you say decontamination or sanitization, you are trying to reduce the pathogenic microbes so that you can deal with that. You can handle those particular instruments without any worry for acquiring the infection. Decontamination is almost equivalent to disinfection because you use disinfectants to decontaminate items. But these are different terms which can be used by individuals. So when you say decontamination, it is almost like saying disinfection. But you also have physical decontamination. For example, you have used an instrument for surgery, and the instrument is soiled with organic matter, blood, tissue, etc. So you would physically decontaminate it by washing, by scrubbing the area, that is physical decontamination. And then you expose those surfaces to certain chemicals, that becomes your chemical decontamination or sanitization, right? So I'm just explaining certain terminologies that you are likely to come across. Now I have written here some organisms in a manner. You have prions, you have cyst, but certain parasitic cysts, specifically certain cysts like cryptosporidium. You have bacterial spores. You have mycobacterium, tuberculosis, or leprae. Then you have lots of other parasites which produce cyst. Then you have viruses, and the size of the virus and presence or absence of envelope on that particular virus. So you can see small enveloped, non-enveloped viruses. then you have trophozoites, then you have gram-negative bacteria, then you have fungus, then you have large non-enveloped viruses, gram-positive bacteria, and last is your enveloped virus. What do you think this is talking about? What do you think this ascending or descending order is talking about? No idea? So here we are talking about the resistance of microbes and the microbial forms. Yes. So it is talking about ability to resist certain sterilization processes. So if you know that I have treated a patient of prions, some CNS surgery has been done in a case of suspected disease, where you are suspecting presence of prions, then that particular instrument is going to be the first to make it sterile. Because prions are high, they demonstrate highest resistance to killing. But organisms like, HIV virus, hepatitis B virus, which are enveloped viruses, though we are so much afraid of them, but they are the most liable to, get killed very, very easily. Exposure even to alcohol wipes is sometimes enough to inactivate these enveloped viruses. So you have to keep in mind that organisms do demonstrate a variable amount of resistance. And the number of organisms, they also influence. So microbial load on a particular article or a surface is going to ensure whether it is going to be easy to disinfect or sterilize, or it is going to be difficult. And nature of organisms you already have seen. Then some other factors which influence the ability of an article or instrument to get sterilized is the chemical agent and the temperature that you are exposing them to. So concentration, when you are learning about disinfectants, you will have to remember what concentration of that particular chemical is going to be effective. And if the load is expected to be more, probably you will have to increase the concentration, or you have to increase the time of exposure. So concentration of the chemical agent, or when you're using heat, then the temperature is going to be important. So nature of the sterilant or disinfectant, every chemical is not going to be effective at the same concentration. Every heating method is not going to be effective at the same temperature. So the nature of that whole process is going to influence your disinfection and sterilization. The time to exposure, temperature already said, and the environment. For example, heat kills more readily at an acidic pH. So the temperature as well as pH are going to be important factors. And remember, I think you know what biofilms are. Anybody who can inform me what biofilms are? Any idea about biofilms? What are biofilms? Unmute and say, what is a biofilm? You have studied morphology of organism, right? So in that morphology, did you read something like glycocalyx, capsulated organisms, and organisms which produce slime? So there are many organisms which produce a very slimy substance, which is not very, slime producing bacteria. So these slime producing bacteria, they are hidden. And it is not very easy for disinfectants to cross these slimy layers, protective layers. So any organism which has got the capacity to produce these slimes, they tend to be very difficult to get disinfected. So you have to really scrub these slimes before the article or the item can be subjected for sterilization or disinfection. So when we are talking about this sterilization disinfection processes, we have to keep in mind that different locations in hospital will have different uses of these sterilizations. So for example, in microbiology laboratory, we sterilize the culture medias. We disinfect the working platforms. You must have read about the biological safety cabinets. They all need to be ensured that they are not they are disinfected and cleaned or decontaminated time to time. So you have to clean the autoclave, sorry, not autoclaves, the incubators. So all these things are the locations where you are making use of this sterilization and disinfection. But in patient care, it is very, very important to provide asepsis, wound care, pre-surgical care, all injectables, all anything which is parenteral, you have to ensure that sterility is maintained. And I think when you talk about disinfectants, at the end, you will also come across Spalding's classification, which helps you in deciding what kind of sterilization or disinfection is required for a particular item. Because there are so many things that are used in patient care. They can be as instruments like, only a BP instrument, thermometer, but you have injectables, you have bed pans. So there are so many things of diverse use which can be categorized depending upon the type of exposure that occurs through those items. And then you have to take a decision whether this particular item needs to be completely free of microbes, or it is okay if it can be only decontaminated and washed properly. So use of sterilization is going to be quite varied. And it is not only in healthcare where you are making use of sterilization and disinfection. You have lots of other industries. Particularly all research industries will ensure that the microbes don't interfere. Particularly drug industry, food industry. So sterilization and disinfection has got a very, very wide application. And depending upon what is the purpose for which this process is being carried out, what kind of material is going to be sterilized or disinfected, what are the likely microbes expected in that particular instrument or item, you have to choose what is going to be the best method. So if you have to classify sterilization method, then you can broadly classify them like this. The crudest or the oldest of all methods, if you see, is sunlight. Because how do you think will make things sterile? Can you share? All of us do like to sit in sun rays. Okay. So Raj is saying you have UV rays which are going to be directly damaging to the genome or DNA of the organisms. And obviously heat itself has got the capacity to kill, because heat leads to drying and desiccation of organisms. So sunlight and drying, these are very, very important basic methods. But the problem is, what is the problem with these methods? Why we don't use them in patient care or in industry? Yes. Why we can't use them or rely on them? Would you rely on an, yes? What do you want to say? We have to take precautions and it is not accurate. Okay. Precautions means what? Okay, sunlight is not always available, so you can't rely on the availability of that thing. And the most important thing is there is no control. You don't really know what was the exposure. Fire safety. What exactly do you mean? Ratnam, can you please clarify your query? You mean to say we can use heating equipments in hospital setting? It is not going to be very feasible and not a very controlled method of ensuring safety of equipments. Yes, that is what probably you are trying to say. Good. So you don't really rely on sunlight for many reasons. Because it is going to be so variable. There are so many factors in the environment, the dust, the pollution, which is going to influence the amount of heat and the exposure to UV rays. So the direction and the environmental factors are going to be very, very uncontrolled ones. So sunlight and drying, though they can kill the microbes, cannot be used in a controlled setting. So we don't rely on them. Heat, heat has got uh, lots of applications when we are talking about sterilization. And broadly dividing, you can have two types of heats. One is a dry heat, another is moist heat. Heat is one of the most commonly adopted method for sterilization. So sterilization by dry heat and sterilization by moist heat is something which you will have to study in some detail. So when you talk about dry heat, we are talking about flaming, incineration, and hot air oven. When we talk about moist heat, then depending upon the temperature which is being used, it is going to be moist. So you have to have water inside. So temperature below 100 degrees centigrade means you are using techniques like pasteurization or directly using water bath where the things are kept, or a specific process of inspissation. At 100 degrees centigrade, it is simply boiling, steaming, and a technique which is termed as tyndallization. Just like inspissation, you have tyndallization, but the temperatures are different. The basic principle of both these inspissation and tyndallization is same, but exposure of that particular item or instrument is at different temperatures. And the most important or the most commonly used system, that is autoclave, works at temperature more than 100 degrees centigrade, and this is under pressure. Right? So every one of you, please read about heat as a method of sterilization in some more details. Then other physical methods of sterilization include filtration. Filtration is very important in terms of removing the microbes, and obviously you will know that this is going to be useful for liquids. And you have different type of filters depending upon what kind of liquid is going to be made free of microbes. And the pore size of these filters will decide what can be retained. The material of these filters and the pore size is going to be very, very important. Right? Then you have radiations, both ionizing as well as non-ionizing radiations can be used. So your X-rays, cosmic rays, gamma rays, they are going to be ionizing radiations. But we know that out of these, you do have gamma radiation as a commonly used method of mass level sterilization. And so all your pre-packed materials, if you see, you will see them as gas sterilization being used. That is your ethylene oxide, ETO sterilized. If you come across any of the pre-packed material that is used in patient care, like catheters, needles, syringes, you just see how that particular thing has been sterilized. So either you will come across gamma radiation or you will come across ETO sterilization. So that is a gas sterilization. Here we are talking about heat and other sterilization methods. And then you have ultrasonic vibrations, which can also be used. Okay. So these are the common methods. Gas sterilization and all I have not really mentioned here. Hmm? So physical methods. These are the physical methods that you have come across. Sunlight, you know how it kills. Drying, you know how deleterious it can be for the organisms if 70 to 80% of their water content is destroyed. Hmm? Dry heat, and moist heat. How do you think is going to be the difference between killing by dry heat and moist heat? You can read. Dry heat kills the microorganism by charring, denaturation of bacterial protein, oxidative damage, and by the toxic effect of elevated levels of electrolytes. So it is going to really denature the protein and is going to produce oxidative damage. Moist heat is going to again do similar kind of things, but also does coagulation of proteins. Now, out of these two heat that we are going to use, which one do you think is going to be a better option? Which one is going to be more effective in terms of killing, dry or moist? Okay. Why moist, Ratnam? Why do you think you are correct? Moist heat is going to be more effective in terms of killing. But why? Yes, please make an effort. Why is moist heat better than dry heat? One of the most common question that is being asked in Biowas. So do you think denaturation is more, but we have denaturation of protein. So that means both are going to denature. You can see here, dry heat is also denaturing and moist heat is also denaturing. What is this? Latent heat and how does it influence killing? You are right. Very good. Why do you think? Yes, please make an effort. Okay. So I think you have said very correctly. When we are using moist heat, we get an additional advantage of what is termed as latent heat. So that makes it very, very effective. And when you say latent heat, I think you understand what it is. So you can see dry heat acts by denaturation, oxidative damage, and toxic effect. It works when the moisture is not there in the environment. But remember, some bacteria and proteins, they are very resistant to this heat, dry heat kind of thing, because they need the energy to oxidize them is requirement is very high, and oxidation kills them. It takes longer time to kill in comparison to coagulation of proteins, right? So moist heat acts by denaturation as well as by coagulation of proteins. So moist heat can act on these bacterial proteins as well, which are very difficult to be killed by oxidative damage. So only denaturation will not work, coagulation will work better. And as very rightly mentioned by Ratna, you have this latent heat which gets liberated when the steam gets condensed on the cooler surface of the article which is being exposed for sterilization. So the temperature increase in the temperature is very fast. So you have moist heat that works much better in comparison to dry heat, and it becomes easy to achieve the thermal death point. So when we talk about thermal death time, you have to remember that it is the time which is required by an organism to be, completely killed, right? And this is influenced, as you have said, the thermal death time is influenced by lots of factors. So presence of organic substances, proteins, nucleic acids, starch, gelatin, sugar, fat, oils, they all influence your thermal death time. So that's why you have to remember in patient care, whenever you are using any instrument and you're putting them for sterilization in any of the instruments, let the instrument or the item be completely clean of all the organic material, extra proteins and contaminants. So that's why thorough washing and cleaning of the surfaces is a must before you expose them into these instruments. Either it is ETO or gamma radiation or autoclaving or hot air oven, you have to ensure that the instrument or item is completely clean to ensure that they get killed within the stipulated time. And presence of disinfectants and higher alkaline acid can increase killing, so it can be adding efficiency into your system. And this you already know. So sterilization time you have to maintain and keep this sterilization time to the point. Every instrument you will when you read, you will come across that this particular instrument has to have this duration of holding period. So this holding period is the sterilization time which is required. And remember, this time is after that particular temperature or the condition of sterilization has been achieved. Once you raise the temperature to, for example, hot air oven needs 160 degrees Celsius. So the time taken to reach to 160 degrees Celsius is not going to be counted in your death time. It is after the temperature has been achieved. You have to ensure that the hot air oven runs for 1 hour or 2 hour, whatever is the time that has to be ensured, right? So keep this concept in mind. Dry heat sterilization techniques, you use flaming and incineration. When you come to laboratory or when you work in microbiology laboratory, you will see that you have those straight wires and loops that we saw. Or we work with the liquid media, we work with culture plates. So flaming is not going to be used in patient care directly. It is used in laboratories to make those wires and loops red hot by putting them in the Bunsen burner flame. So you burn them till they become red and then cool them. So that is considered to be a quite effective method of sterilization. Test tubes, you can flame, you can just pass the mouths of these test tubes which are being used for inoculation to flame them and ensure that they are disinfected. You're not going to make them sterile, but you pass through the flame 2 or 3 times. You pass the slides through the flame 2 or 3 times just to ensure the microbial load is reduced. And I'm sure you know what incineration is. Have you seen incinerator that we have in the hospital? Anyone? Just in case you have not seen, when you come, please do visit the site where we have incineration. Okay, so you have seen that now. Good. So you see the incinerator and see how it works. But Remember, incineration is not a method of sterilization. Hmm? Incineration is a method of final waste. Most of the times you do make this mistake of differentiating incineration and inspissation. There are two different things. Incineration is a method where you dispose the biomedical waste final thing that you have to do. So it completely burns. So the waste is sterilized, but that does not make it user friendly for direct patient care. Hmm? The temperature of incinerator is very high. You can see 870 to 1200 degrees centigrade. So whatever is there in the biomedical bag waste goes for incineration. So that contains all your body parts, microbiological waste, dressing material, all those kind of things will go for incineration and it will be converted into ash. And you have certain toxic gases that get released under this exposure of high heat. So you ensure that you are not incinerating anything which is chlorinated in nature. So all your plastic waste will never be incinerated. If it is a non-chlorinated plastic, you can send it for incineration. But just to be on safer side, we ensure that no plastic is sent, because we have those dioxins and fluorans, all those gases that are released. Okay, so that is flaming and incineration. The most common method that we use in patient care is hot air oven. That is a dry heat sterilizer. So you have these hot air ovens with the metal cupboards inside. And in this hot air ovens, you have them heated through electricity. And there is also a fan inside so that the heat gets distributed completely. Heat keeps rotating inside for equal distribution of heat. And it also has got a temperature control or monitor. So you have a thermostat. So this thermostat will ensure that the set temperature is maintained and you do have a timer for a particular time duration. Okay. So you remember the time and the temperature of a hot air oven. You have to ensure that your material is exposed for 160 degrees centigrade and you keep a holding time of 2 hours. Please remember temperature and time of hot air oven autoclave specifically. Okay, so what are the things that you can sterilize in hot air oven? All your glasswares can be very easily sterilized. So all microbiological raw materials that we prepare in these petri dishes, flask, pipettes, they can be easily sterilized in hot air oven. All your surgical instruments like scalpel, forceps, etc., the metal ones can be sterilized. Chemicals like liquid paraffin, fat, glycerol, oil, glove powder, etc., can also be sterilized in hot air oven. Certain precautions that you need to take. You obviously any instrument must not be overloaded because the circulation of heat is going to be compromised and that will result in compromise of your process. Then material should be not arranged in such a manner. You can't stack one over another. They have to be, each item has to be kept separately on the platforms inside, so that the free circulation of air is ensured. And as we said, for a dry heat, this is a dry heat, so you have to avoid the presence of moisture. So ensure that all your items are completely dry. If you are using test tubes and flask, ensure that they are plugged, otherwise they might crack. You wrap the items. All your glass items need to be wrapped properly and any inflammable material must not be kept. You can keep silicone rubber inside because it is not inflammable, right? And once the time is over, you don't open it immediately. You have to give a cooling period, otherwise all your glassware will break. Because the change is to ensure that your material is properly sterilized, you use sterilization control. And in sterilization control, you have both biological indicators and certain, what do you say, the physical control measures, right? So physical control means you ensure that your temperature is set correctly, you are giving enough time. So your temperature monitoring becomes important, and you have to time check that the thermostat is working well. It shows 160 degrees centigrade, means the temperature inside has to be 160 degrees centigrade. So this has to be done properly by maintaining the equipment. Once the cycle is run, along with the load, you put the strips which contain the spores of Bacillus atrophius. Initially, we used to have spores of Clostridium tetani, but now Bacillus atrophius spores are used. And these spores must get killed on exposure to the designated temperature and time. So that becomes important. Once you expose these spore strips to hot air oven, then you take them after the cycle and inoculate them in culture medium. So if the spores have killed, they will not grow, and your no growth will indicate that the spores have been killed. So that becomes your biological control. So this is about hot air oven. So what are the things that we have talked about? Quickly. Then I move on to moist heat. In moist heat, I will say one or two methods, and autoclave we will learn in the next class. So moist heat sterilization, dry heat sterilization, everything is clear till now? Any query? Okay. So if there are any queries, please feel free to ask. So in dry heat, we have learned flaming, we have learned incineration, we have learned hot air oven. In moist heat sterilization, in the table where classification was shown, I had said, depending on the temperature, it can be less than 100, at 100, or more than 100 degrees centigrade. So less than 100, the most common technique which is being used is pasteurization. And we all know being used. You know that all your Amul milk bags, they are pasteurized. So for dairy products, it is very, very commonly used, right? But it can also be used for other beverages. You can have juices, beers, fruits and vegetables pasteurized. When you are talking about pasteurization, you can pasteurize by two different methods. One is termed as a holder method, another is termed as a flash method. So in holders, cigarette, you hold that temperature for 30 minutes. And in flash, you raise the temperature to 72 degrees centigrade and keep it only for 20 seconds. And then immediately this rapid change in temperature leads to killing of the microbes, and this can kill most of strong considered the organisms which have been considered quite strong, like mycobacteria. Pathogens will get killed. Your mycobacteria, brucella and salmonella will also get killed. But certain organisms like Coxiella burnetii using flash technique of sterilization. So this is pasteurization. Remember, very common for milk and milk products. Then in laboratory, we do use water baths, particularly if you have to disinfect body fluids, serum, and vaccines. 60 degrees centigrade for 1 hour. Put them in a water bath where the temperature is maintained at this particular time and duration. Serum or heat labile fluid can also be disinfected at 56 degrees centigrade if they are maintained for 1 hour. So water bath can also be used. Then inspissation, fractional sterilization, right? So fractional sterilization means you are heating an item on 3 successive days. The temperature is 80 to 85 degrees centigrade and you keep that for 30 minutes. So what is going to happen over a period of 3 exposures? On first exposure, all your vegetative organisms are going to be killed at this particular temperature and time. Then the time in between is going to stimulate the spores to germinate. So the spores will become vegetative and the frequent exposures will kill these germinated vegetative forms. So on 3 successive days, you give time for germination of spores and keep killing the vegetative forms. So this is a very commonly used method for sterilization of your LJ medias, egg containing medias and serum containing medias. Because high temperature will result in coagulation of proteins. So you use intermittent or fractional sterilization. So less than 100 degree, it is your inspissation. Then at 100 degrees centigrade, you can boil any item, very commonly used for household items, but not really well controlled. So not commonly used nowadays. Then you have Arnold's steam sterilizer. This Arnold's steam sterilizer is useful for media which get decomposed at temperatures of your autoclave. You are exposing the item to steam at 100 degrees Celsius. So this leads to killing of your vegetative cells, but of course not your spores and heat resistant organisms. Last is your tyndallization, heat at 100 degrees centigrade. So if you can see, this is similar to your inspissation, but here you are using 100 degrees centigrade for 20 minutes, 3 consecutive days. Okay, so only the difference is temperature, and it is used for sterilization of medias. So this is at 100 degree centigrade. Heat above 100 degrees centigrade is your autoclaving, and because autoclaving is very, very important, so we will start this fresh in the next class. Is that okay? Any queries till now? Hello. No queries? Okay. So in 2 minutes, write down whatever we have learned till now. And I'll ask someone to read what you have written. 2 minutes. Write down what you have learned in today's class. And then we can finish.